Today, we're going on a journey. We're about to explore a hidden universe that's all around us and, yeah, even inside of us. We're unlocking the microscopic world and the story of the cell. So, let me just start with a question for you. What if the world you see every day is just the tip of the iceberg? What if there's an entire teeming reality that you are completely, totally blind to? Well, there is, and it all comes down to this number, 0.2 millimeters. That's it. That's the absolute limit of what your unaided eye can resolve. Anything smaller, any two little dots closer together than that, they just blur into one. It's a physical barrier to our perception. And for almost all of human history, that universe of things smaller than 0.2 millimeters was a complete and utter mystery. Think about it, an entire dimension of life just existing in parallel with our own, totally invisible. I mean, really let that sink in. Every single drop of water, every speck of dust, held secrets we couldn't even begin to imagine. This world was always here, just waiting for someone to find the key to unlock it. So, who were the first explorers of this new frontier? Let's meet the pioneers, the very first people to peer through the looking glass into this microscopic world. It all kicks off back in the 17th century. So in 1665, a scientist named Robert Hooke takes a look at a super thin sliver of cork under his microscope, which was pretty primitive back then. And he didn't see a solid mass. He saw thousands of tiny, empty boxes. Then, just a few years later, this guy Anton von Leeuwenhoek uses a way better microscope. He looks at a drop of pond water and is just floored. It was filled with these tiny, swimming creatures. Life, right there, where no one thought it could possibly exist. And what did Hooke call those little boxes he saw? Well, he called them cells. You know why? Because they reminded him of the small rooms or chambers that monks lived in. Such a simple name for a discovery that would literally change everything we thought we knew about life itself. Okay, so, with the door to this incredible new world thrown wide open, the next big challenge was to actually understand it. Scientists started to realize they weren't just looking at random specks. They were observing the very foundation of life. They needed a rulebook. And that rulebook became known as the cell theory. Think of it like the constitution for all life on Earth. It lays out the fundamental laws that govern every single living organism. No exceptions. And these are the 3 laws that just broke biology wide open. First, if it's alive, it's made of cells. You, a tree, a tiny bacterium, all of it, cellular. Second, the cell is the smallest thing that can truly be called alive. It's the fundamental unit. And third, and this one is crucial, cells don't just appear out of thin air. Every single cell comes from another pre-existing cell. It's an unbroken chain stretching back to the very, very beginning of life. But look, to really prove these rules and to explore this tiny universe, scientists needed better tools. The story of the cell is also the story of the technology we built to see it. It's this constant race to make the invisible visible. So back to our barrier. How do you get past that 0.2 millimeter wall? The answer is pretty straightforward, right? You magnify it. You have to find a way to make these tiny things look big. Now, the magic of what we call a compound microscope is that it combines lenses. It's actually a simple bit of math. You just multiply the power of the eyepiece, that's the part you look through, with the power of the objective lens, the one down by the specimen. So let's take a really standard setup. A 10x eyepiece combined with a common 40x objective lens, well, that gives you a total magnification of 400 times. All of a sudden, what was just a blurry dot is now 400 times larger, with all of its incredible hidden details just snapping right into focus. And this idea led to this super rapid evolution of the tool. We went from simple single-lens microscopes to these powerful compound ones that could magnify up to 2,000 times. But to go even further, to see even smaller, we had to abandon light itself. And this really shows you the sheer scale of that leap. A compound microscope, it uses a beam of light. But an electron microscope uses a beam of electrons. And because electrons have a much, much shorter wavelength, they can resolve these unbelievably small details. I mean, just look at that jump in power. We go from maybe 2,000 times magnification to over 1,000,000. It's a completely different league. And what does that incredible power unlock?
Well, it allows us to go from just seeing cells to seeing the actual machinery inside the cells. This is a journey into the truly unseeable world.
You see, with the electron microscope, we weren't just making a catalog of different kinds of cells anymore. No, we were entering a whole new dimension of biology, seeing the fundamental components that actually make life work.
For the very first time, we could get a sharp, clear image of a virus. This strange thing on the very border of what's alive and what's not, way too small to ever be seen with a light microscope.
But we could push it even further. We could see the very molecule of life itself, that elegant double helix of a strand of DNA, the blueprint, right there in front of us.
And finally, the ultimate destination of our journey inward. With the most powerful electron microscopes today, we can actually resolve the image of a single atom, the fundamental building block of, well, everything. We went from seeing a monk's chamber to seeing the very atom that makes up the wall of that chamber.
I mean, what an incredible journey, right? From Robert Hooke seeing simple little boxes and a piece of cork, to us, today, being able to visualize the very atoms that build those boxes. The history of science has shown us time and again that every time we build a new lens to see the world, we discover a new universe we never even knew existed. So the only real question is, what are we going to see through the next one?